Hi friends. Today we are going to discuss about color coding of blood collection tube and their uniqueness with order of draw. The containers used for blood storage is separated by an unique color. This denotes their anticoagulant. Anticoagulants are the chemical compound that has the capacity to block the coagulation process and keep the blood in a liquid form for longer time. Normally, if the blood comes out of the body, that will clot within 8 minutes, but when anticoagulant added there will be no coagulation on that particular sample. Hence the blood coagulation occurs against activation of 12 factors in a proper order. When we use anticoagulant, the factor will not be activated, so the blood clotting will not occur. Coagulation mechanism and their factors will be discussed later in biochemistry part of our channel. There are a lot of blood collection tube color coding available. Among them here we are going to discuss about more important tubes. 1. Blood culture aerobic. 2. Blood culture anaerobic. 3. Plain. 4. Clot activator. 5. Sodium citrate. 6. Heparin. 7. EDTA, K2, K3. 8. Sodium fluoride and potassium oxalate. 1. Blood culture aerobic tube is in blue color cap. And, this is used to identify the presence of aerobic bacteria in that particular sample. All blood culture tubes are containing anticoagulants along with growth medium that supports growth of those bacteria. 2. Blood culture anaerobic tube is in violet color cap. And, this is used to identify the presence of anaerobic bacteria in that particular sample. If the child is pediatric, the sample must taken in the pediatric culture tube, which is in violet color coding. 3. Plain tubes and clot activator tubes are red in color. No anticoagulants and clot activators are present in those red tubes respectively. That is the only difference between these two tubes. Sometimes yellow cap tube is used instead those red tubes. That contain gel inside it which separates serum and cells automatically after centrifugation. All these three will separate serum from whole blood. Commonly plain and clot activator tubes are used. Gel tubes are not commonly used even though it simplifies our work by separating serum automatically after centrifugation. Because the gel present in the tube interferes with some test procedure. 4. Sodium citrate is used as an anticoagulant in two tubes, one for coagulation profile tests which is in blue color and ESR tube which is in black color. Both these two are having same constituents, but the only difference is concentration and dilution. Coagulation profile tubes containing 3.2% sodium citrate and diluted with blood in the ratio of 1:9. And, ESR tubes are having 3.8% sodium citrate and diluted with blood in the ratio of 1:4. This anticoagulant inhibits the coagulation process by chelating the calcium. Unlike EDTA, this is weak chelator. So the chelating process is reversible. 5. Heparin is an anticoagulant which is normally in our human system. In this tube the same anticoagulant is going to be used. This tube commonly used instead red clot tube. Unlike clot activator, this tube gave plasma instead serum. Many tests are common for clot activator and heparin tube. 6. Commonly two types of EDTA tubes are used, one is K2 EDTA and another one is K3 EDTA. Most commonly, K2 EDTA is preferred, for counting of blood cells and other hematological studies. Hence, the K2 is sprayed inside the walls of tubes. But, K3 tube contains liquid. So the dilution problem will lead to 1 to 2% error in report and quantification studies. As we discussed in sodium citrate tubes, these anticoagulant also inhibit calcium by chelating calcium. Unlike sodium citrate, EDTA is not reversible. 7. Sodium citrate and potassium oxalate tubes are introduced for the quantification estimation of glucose. Normally, the sample taken from other than this tube will be decreasing time in glucose concentration. Due to oxidative process, sodium citrate will inhibit the oxidation process, and this tube will be used when the sample is not used immediately and stored after collection for more than 15 minutes. Upon this list, some anticoagulant tubes are available for special tests. But common labs must contain these tubes. The tubes cannot be filled in any order. After collection there is an order for filling the tubes. If more tubes are used, that is called as order of draw, which is given here. This order of blood draw is used to prevent the sample from cross-contamination of additives and others, between various sample container. When anticoagulants used in the tube must be inverted for particular time for mixing of sample with anticoagulant. The inversion is the process is explained in this picture. Tubes and their inversion recommendation is given here. Any healthy suggestions and feedback, comment. If you like this video, hit the like button and share with your friends. Do subscribe and click the bell icon if you want the same type of videos immediately after updating my channel. Next video will be about centrifugation and separation of blood samples. Thank you for watching.